Okay, so thank you everyone for joining us and Happy New Year. We're excited to restart the last second half of the seminar series. Many of you may remember Dr. Thomas Quail from his time in Montreal. He completed his PhD at McGill University in the labs of Dr. Leon Glass and Dr. Alan Schreiber. And he was an integral member of the student arm of CAMBAM, and that's how we met, probably 10 years ago, Tom, something like that. So in his PhD, Thomas worked on how to predict the onset of abnormal cardiac rhythms using nonlinear dynamics and reconstituted cardiac systems. And he's now a postdoc in the, I'm not sure I'm going to say this right, Bruges?
Yeah, Bruges.
Okay, Bruges lab, sorry. And he studies the physical principles of genome self-organization using a combination of single molecule biophysical approaches and reconstitutive biochemistry, xenopus egg extracts, and theory. So outside of the lab, he walks his dog Birdie, who's a street dog from Romania who resembles a miniature German Shepherd. So that's very important information. We're glad that that was included, and I'm really excited to hear Thomas's talk today. So please go ahead, Thomas.
Great. Well, thanks very much for the nice introduction. It's exciting for me to give this talk. So I know this is I know it's a more math-heavy series. I checked some of the older seminar series, but this will be. There'll be a lot of experiments, and then there'll be a bit of theory taken mostly from statistical physics. And so I think there'll be a bit something for everyone. And so today I'm going to talk about experiments that we've been doing in the lab. So this is in the lab of Jean Bruges at the Max Planck Institute of Molecular Cell Biology and Genetics, related to understanding how collections of proteins can generate forces on DNA. So in every cell in your body, there's a cell nucleus. And within the cell nucleus, the nucleus houses the genetic material of that cell. And so this is a problem because if you're to stretch out the genome in a linear sense, it'll stretch out to be about 2 meters for a single cell nucleus. But this needs to be packaged and organized into a sphere, essentially, with a diameter of roughly 10 microns. And so we know a little bit about how the genome is organized. And we know that the genome is organized across different length scales by proteins, actually. And so here's a nice schematic taken from a recent paper from Tomaselli, where you have the DNA helix, which is wrapped around at very short length scales by core histones. And at intermediate length scales, this nucleosomal DNA is turned into compartments called chromatin loops, and that's done by actively extruding motors. And then at even longer length scales, these chromatin domains or loops are further packaged into chromosomal territories. And so over the last few decades, there's been a ton of work that's gone into identifying the proteins associated with this genome organizations through fantastic genetic, proteomic, and structural biology studies. So structural biology is going into the precise near atomic resolution details of each of these proteins. But I would propose that we don't really have a good understanding of how all of these individual proteins are collectively working together in space and time in order to give rise to this organization across different length scales. Now, so this leads to a question of how microscopic units come together to give rise to patterns and dynamics. And this is a general problem with many examples within biology and beyond. And so here are a few examples. So again, you can see the collective behavior takes place across different length scales. So here's an example of microtubules giving rise, coming together via the activity of motor proteins and passive cross-linkers to give rise to a spindle reconstituted in xenopus egg extracts. And so the mitotic spindle is responsible for separating the chromosomes through cell division. Another example is that cells have to work together within a developing zebrafish embryo in order to give rise to proper embryonic development. And at even longer length scales, cells of different cell types have to be in the right place at the right time in order to perform physiological functions, such as the beating of a heart. So my approach, I thought that it would be good to actually tell you a bit about my approach to trying to understand this problem. So my approach is basically trying to reconstitute. So how do you try to understand collective behavior? So my approach has been, you can reconstitute these more complex things in a simpler fashion experimentally. You can quantify them, and then you can apply theories taken from nonlinear dynamics or statistical physics to try to understand these patterns. And so I got really, I wanted to just talk briefly because I'm talking about, it's a Montreal crowd, but this approach is really, I guess, driven into me as a PhD student in the labs of Leon and Alvin, and where I basically employed this approach to try to understand how to predict the onset of abnormal cardiac rhythms. So this is a trace from a person who had a normal sinus rhythm in the afternoon, and later on that evening, there's a transition from this normal rhythm to a ventricular tachycardia or an abnormal cardiac rhythm. So how do you try to understand these transitions in complex dynamics? And so in Leon and Alvin's lab, the approach was to reconstitute the beat patterns using spontaneously beating aggregates of embryonic chick cells or monolayers of these cells. And then you can go about quantifying the dynamics. So here is a trace of the interbeat intervals as a function of time following the treatment with a potassium channel blocker from one of these spontaneously beating aggregates. You can see that there's a change in the beat patterns over time, over tens of minutes timescales. And then you can apply theories taken from nonlinear dynamics. And one thing that we did was to look at these period doubling bifurcations that were induced by the treatment of a potassium channel blocker, and try to understand when the system was in the neighborhood of this period doubling bifurcation, whether we could predict how far the system is from transitioning to this abnormal alternate pattern. And so in my postdoc, I wanted to use this approach in a different context. So the overarching problem that I'm interested in is understanding what physical mechanisms organize the genome. So we know the content of the proteins that are giving rise to this organization, but what's the underlying physical principles that give rise to this compartmentalization or organization? And so again, this organization is taking place across different length scales. We know that at a tens of kilobase per length scale, one example of a mechanism that's relevant is a condensation pattern. At tens of kilobase per to megabase per length scale, loop extrusion by actively extruding motors such as cohesin and condensin can also give rise to this genome organization. And then at even longer length scales, at the length scale of chromatin compartments, polymer-polymer interactions between heterochromatin and euchromatin could also be relevant. And so for the rest of my talk, I'll discuss protein condensation and how collections of proteins work together to bring distal DNA regions together. And so this work is really rooted in a problem related to transcription. And so for proper transcriptional control, proper gene regulation, enhancers which are small strips of DNA, must be brought into close proximity to a gene promoter in order for that gene to be turned on. However, there's a spatial problem, which is that in the linear genome space, these enhancers can be located far from the promoter of that gene body. So the question is, how do individual proteins within the transcriptional machinery, here given in orange, how do they work collectively together to bring this enhancer close to this promoter? And so a few years ago, it was posited that a number of papers came out with a very provocative hypothesis, which was that biomolecular condensates could drive the formation of these transcriptional hubs, or bringing enhancers and promoters together. And so I'm going to spend just the rest of this slide and another slide to try to give you all an intuition about what I mean for what a biomolecular condensate actually is. And so I think the best way to think about it is how does the cytoplasm and the nucleoplasm of a specific cell, how does it organize its biochemistry? So one way to organize its biochemistry is using membranes. So for example, in the nucleus has a double lipid bilayer membrane. And that's a way of sequestering certain factors into the nucleus and then keeping some factors in the cytoplasm. So that's one way. But it was actually discovered in the last 10, 15 years that membraneless compartments are also a way that the cell uses to organize its biochemistry. And so there's now been a number of examples over the past 10, 15 years that are relevant in terms of understanding the cellular biochemistry. So for example, in the cytoplasm, when a cell is in a stressed condition, it gives rise to something called a stress granule. So this is a membraneless compartment that sequesters RNA and RNA binding proteins that are related to the stress response or cell survival. And in the nucleus, for example, the nucleolus has been proposed to be a membraneless compartment within the nucleus, where certain activities can go on. Transcriptional hubs are also another example, such as well as paraspeckles and nuclear speckles. And I'll just because it's a key part of the talk, I'll just spend a little bit more time on it. So maybe to give you a bit of a flavor, I've included some videos. And so this is a paper from '09. So this is kind of one of the classic references. And these are P granules in the one cell stage of a C. elegans. And what's labeled is a marker for the P granule, so PGL1. And you can see that, so this is just cytoplasm. And you can see that there's a number of these speckles that they're on the order of maybe 3 to 5 microns. And they're circular, and they're moving around, and they're fusing, and so on and so forth. And so when people have started looking into the content of these biomolecular condensates and how they form, they realized that they could form in a way consistent with liquid-liquid phase separation. And so you can also induce them. So this is an example of a HeLa cell that there was an injection of RNAs. You degrade the RNA, and you can see the formation of these. So this is, I would say, more an extreme example where you're inducing the formation of these things. And then you can also take purified components. And you can take, this is GFP FUS here. This is purified GFP FUS. And then you can image these proteins, and you can see that they're fusing in a way that's very consistent with how a liquid droplet would fuse in the context of oil and vinegar, for example. And it's an area of very active research right now, because of course the cytoplasm doesn't necessarily have two components like you have here, oil and vinegar, but rather it's got tens of thousands of different proteins, and some of those proteins have post-translational modifications, and so you have a very complex fluid. And so figuring out the role of these, of how liquid these things are is an area very much of active research. So I hope that gives you all a flavor of biomolecular condensate information. So I'm going back now. So we were thinking about this problem, and we realized that in principle, biomolecular condensates that are interacting with chromatin can indeed generate force. And so we know that because from physics, we know that a liquid that's interacting with a solid generates forces. And those forces are what accounts for how liquid droplets can maintain the tension on a spider web, for example. And it's also relevant for understanding how these forces are relevant for understanding how certain insects can also walk on water. So we asked three specific questions. One, are these condensation forces relevant? So are they generating forces that are relevant in the context of DNA or chromatin? So are we an order of magnitude off? Yes, it's generating force, but it's not actually doing anything. The second point is, can we extract numbers for the physical parameters associated with this process? And then the last question is, how does the protein biochemistry of these individual proteins govern these forces? So that's what we set out to do, and I hope that I can answer these questions by the end of the talk. So we developed an assay to image single molecules of DNA and a single transcription factor FOXA1. So our approach was, this is getting again to the reconstitutive side of my approach. So this is a very complicated process, and so we wanted to reduce the complexity as much as possible, such that we could try to understand these questions. So the first thing we did was we developed this assay where we could image the single molecules of DNA. We took lambda phage DNA, here given by this squiggly DNA. We attached biotin to the ends of this linear piece of DNA. And then we attached that to a functionalized cover slip. And I think, so on the right-hand side, the way that we're visualizing the DNA is we used a DNA dye. And I think the key thing to remember here is that these three pieces of DNA have the same number of base pairs, but because they have different end-to-end attachment points, they have different intrinsic or imposed tensions, essentially. So they have different forces that are being imposed on it. So you can see when the end-to-end distance is lower, so this is given in the bottom trace, or the bottom movie rather, a single molecule of DNA can explore much more of its space because it's under less tension. However, when the end-to-end distance is higher, the fluctuations associated with the single molecule of DNA are far more constrained. So it's like think about like an elastic band that you're stretching. And this is important for us because we were thinking about the DNA molecule bending back on itself, thought that tension could be a relevant parameter for understanding this problem. So that's the assay, and then the transcription factor that we were interested in is called forkhead-box-A1 or FOXA1, and all you need to know about this transcription factor. So here's just a schematic of the protein structure. It has a disordered N-terminus. So what that means is just that the N-terminus doesn't have a very well-defined structure, so it's just flopping around a lot. And then it's got a very well-defined DNA binding domain. So that structure of the DNA binding domain doesn't really change. So it can, of course, rotate and translate, but it doesn't change. And then it's also got a disordered C-terminus. And so this is a depiction from AlphaFold, and essentially what I want you to take or extract from the AlphaFold prediction is that you have this very well-defined DNA binding domain given here in blue, and then it's mostly, you can think of it as spaghetti. And so the formation, and another thing to note is that the formation of these biomolecular condensates, these floppy parts of these proteins has been implicated in the formation of these condensates. So you can think about these floppy parts of the protein weakly interacting with themselves or other proteins to form these condensates. Okay. Okay, so we have the assay and we have the protein that we purified, and so what do we actually find? So what we found out was that when you had a piece of DNA that was very stretched out, when you flowed FOXA1, a labeled recombinant FOXA1 into the channel, FOXA1 would bind, it would more or less wet the DNA strand, and then would form these distinct enriched regions of DNA, but of protein. But the DNA would remain largely unaffected. It would just continue to fluctuate as normal, and nothing would really happen. However, when the end-to-end distance was lower, so again, this is the DNA, the number of base pairs here and the number of base pairs here, it's the same. When the end-to-end distance was lower, so you got a floppy piece of DNA, FOXA1 would again adsorb to the DNA surface, but would form this very strongly enriched protein DNA co-condensate. So you essentially you'd have these FOXA1 molecules that would reel in the slack part of the DNA molecule. So you can, of course, take many different examples, and here I have, again, I want to reiterate that these are different examples, but they have the same number of base pairs. So again, if you look at the DNA molecule and you look at a representative FOX condensate, the DNA is not enriched within this FOX condensate. However, as the end-to-end distance is decreased, or the tension on the molecule decreases, if you look at this FOX condensate, for example, this is very strongly enriched with DNA. And you can simply look at you can add up the fluorescence for your DNA, and you can make a one-dimensional profile of the DNA and the FOX, and then simply take a cross correlation, a normalized cross correlation. And so you can see when the end-to-end distance L, here given in microns, is low, the correlation is very high. So you can, again, see when the FOX signal is high, the DNA signal is high. But that correlation drops off to approximately 0 as the end-to-end distance is increased. So taken together, what we found was that FOXA1 is forming these protein DNA co-condensates in a tension dependent manner. Another neat example was we had owing to the fact that the biotin was binding to streptavidin on the functionalized cover slip, and there's multiple biotin attachment points on streptavidin. So what you would get examples where you have two individual DNA molecules that share one attachment point and that have two independent attachment points. And so when we flowed in FOXA1 and we looked at these examples, you could see that first off, FOXA1 was seemingly wetting these DNA pieces, and it would condense them together. So it would form. So you can see here given in the schematic, you share one attachment point. You have two separate attachment points, and then over time, as more FOXA1 is adsorbing to the DNA pieces, you get this characteristic zipped morphology. So we found that FOXA1 is condensing DNA in both cis and trans, but biochemically, how is this working? And so, we did a lot of mutant studies, but I'm just going to basically simplify it to this one slide. And so what we discovered was, in fact, that the disordered C-terminus is what's driving this protein DNA co-condensation. So here is an example where we locked off the N-terminus, the disordered M-terminus of FOXA1, and we can see that we got this strong protein DNA co-condensation. However, when we took off the C-terminus, we got adsorption again, or binding to the piece of DNA. We didn't get this, we strongly inhibited the protein DNA co-condensation. So again, this is was consistent with previous studies that have shown that these disordered regions of proteins are playing a strong role in the formation of biomolecular condensates and their activities. So I just want to step back for a moment to give you a kind of a hand-wavy picture of what we thought was going on, and then I'll jump into the thermodynamic description. So what we think is happening is that when the DNA tension is high, FOXA1 is binding to the DNA. Because of its propensity to self-interact or form these condensates, you're getting the formation of these condensates that's generating a force. Now, if that force cannot overcome the intrinsic DNA tension, then nothing happens to the DNA. However, when the tension on the DNA molecule is lower, or when the end-to-end distance is lower, FOXA1 will again bind to DNA. It'll self-interact, likely through the disordered C-terminus, and this will generate a force. But because that force is greater than the intrinsic DNA tension, this buckles the DNA and essentially reeling it into this protein DNA co-condensate. Now, to more quantitatively describe our phenomenological picture, we wrote down a thermodynamic description of protein DNA co-condensation, which I want to slowly go through with you. And so here is the free energy associated with the generation of a protein DNA co-condensate as a function of L, which is the end-to-end distance, and LD, which is the amount of condensed DNA. And so this free energy term, we divided it up into two. The one was the free energy associated with nucleating a droplet with this amount of DNA, LD. And then the second term is, you can think of it as essentially a work term. So it's the work that the condensate has to generate to pull a certain amount of DNA into the condensate. So I'll just start with the condensate free energy first. So we took the way that we described the condensate free energy was we first looked at the amount of free energy gained or lost associated with nucleating a droplet with radius r. And so from nucleation theory, there's two terms. There's a volume term. And so you can see here that, so negative is you gain free energy. So there's a new term here, which is essentially the condensation free energy gained per volume times the volume of the droplet. And so that for bigger droplets, you gain more free energy, essentially. However, if you're making a droplet, you have to pay free energy to have a surface, essentially. And so here, this gives us gamma, which is a surface tension, times the surface area. Now, to relate the amount of DNA within this droplet to the volume of the condensate, we assume that the volume of the condensate went linearly with the amount of condensed DNA, so alpha LD. And so we could simply plug this into our condensate term. This is the associated condensate free energy with nucleating this protein DNA co-condensate as a function of the amount of condensed DNA. And the work term, essentially, we have a force that you can integrate as a function of the length. Again, you can think about that as essentially the work that the amount of energy, amount of work that the condensate has to perform to pull a certain amount of DNA into the condensate. And so at the end of the day, you get this term again, which is the amount of free energy gained or lost as a function of the end-to-end distance and the amount of condensed DNA. And so to give you a flavor for what you can extract from this thermodynamic description, what's neat is that you can plot the amount of free energy for a given end-to-end distance, and then you can, as a function of the amount of condensed DNA. And so, okay, so here, if the end-to-end distance is 1, that means that the ends are very close together. So the DNA molecule is very, very floppy. And so when the DNA molecule is very floppy, if we look at the minima of this free energy, LD, this is roughly 15 microns. And the total contour length of the DNA molecule is 16.5 microns. So that means that over 90% of the polymer is within this condensate. Now, as you increase the end-to-end distance, or you increase the imposed DNA tension, so here we have end-to-end distance of equal to 7 microns, you can see that the amount of condensed DNA, if you look at the minima of this free energy, is decreasing. So that makes sense. So you can think about it as you increase the tension on the DNA molecule, it's going to be more difficult for the condensate to pull more DNA into the condensate. And what's interesting is that at a very specific end-to-end distance, so somewhere between 9 and 11 microns, there's an abrupt transition in the minima of the free energy that goes from something that's non-zero to something that's 0. So this is reminiscent of an abrupt first-order phase transition. Okay. So that's essentially what you can extract from this free energy. But what I'm interested in is trying to use this free energy to gain estimates for the physical parameters associated with this protein DNA co-condensation. And so the thermodynamic description has 3 parameters, or it's described by 3 parameters. So it's alpha, which is the DNA packing density, nu, which is the condensation free energy per volume, and gamma, which is the associated surface tension. So alpha is the DNA packing density, and that comes from an assumption we made, which was that the volume goes linearly with the amount, the volume of this condensate goes linearly with the amount of condensed DNA. So this is an assumption. So we just went ahead and analyzed that data. And indeed, the relationship was linear. So that means that the relationship between the volume and the amount of condensed DNA is linear. That gives us an estimate for this mu parameter of 0.04 microns squared, which simply comes from the slope of this. Now, in order to obtain estimates of nu and gamma, we generated 2 measurables. So one was plotting the amount of condensed DNA as a function of the end-to-end distance. And so again, you can see, so gray are individual DNA molecules. So you can see when the end-to-end distance is low, that the amount of condensed DNA is high, and then that drops off to 0 at higher end-to-end distances. And then the second measurable is the probability that FOXA1 would condense DNA as a function of end-to-end distance. And what was nice is that we were able to obtain experimental data and theoretical expressions, here given in orange, for both measurables, LD as a function of L or pcond as a function of L. And by minimizing the error associated with the theoretical expression and the experimental data, we were able to obtain estimates for the condensation free energy per volume, the condensate surface tension. So the condensation free energy per volume estimate that we got was roughly 300 kBt per micron cubed. And so we were getting condensates at low tensions on the order of about a micron cubed. So you can think about the amount of condensation free energy gain is about 300 kBt. And the condensate surface tension was roughly 0.1 piconewton per micron. And so there had been estimates for biomolecular condensates that had been formed in bulk, similar to examples that I showed you earlier in the talk. And they were approximately on the same order. And so I talked a lot about this abrupt first order phase transition that came from the thermodynamics. And what was nice is that we also observed this in the experimental data. So this here is the probability that FOXA1 would condense DNA. And you can see that the thermodynamic description shows this, this abrupt transition, which is also consistent with the data. And the critical end-to-end distance that we measured was approximately 10.7 microns of end-to-end distance. And so what was also nice is that using the amount of condensed DNA as a function of end-to-end distance, we were able to obtain a prediction of the force that FOXA1 was exerting. And so here again in gray we have the single strand, single examples. And what we observed was that the FOXA1 condensates are generating forces on the order of below a piconewton. And what's interesting is that it seems that the force is more or less buffered. And it's kind of neat to think, so this buffering effect is also similar to how these liquid droplets are buffering the tension on a spider web. Now, to actually check these forces, you can, so this was simply an estimate from fluorescence, but there's actually a more sensitive way of measuring the forces. And so that's to use an optical tweezer system. And an optical tweezer system, what you can do is essentially trap polystyrene beads with a laser. You can attach the same molecule of DNA that we were using for all of the turf microscopy data that I showed you before between two beads. And so what the experiment that you do is you trap these polystyrene beads, you move these traps into a channel containing DNA. The DNA, a single molecule of DNA will then attach to two polystyrene beads. Then we moved this single molecule of DNA into a buffer channel. We brought the distance between the polystyrene beads to a known end-to-end distance that we know FOXA1 could generate these protein DNA co-condensates. And then we brought this single molecule of DNA into a channel containing FOXA1. And what we observed was if you looked, and so essentially if FOXA1 is generating a force, you can think about one of these beads is kept very, it's a hard trap, so it's not going to move, but the other trap is a softer trap. And so if FOXA1 is going to pull on the DNA molecule, it's going to change the distance between these two beats. And so because because we know the force extension curve associated with this DNA molecule, you can get a relationship between how far the bead is moved with how much force FOXA1 has exerted. And so if you compute the force as a function of time, when there was no FOXA1, so this is just a channel that has buffer, the force remains approximately 0. So the FOXA1 does not exert any force. However, when it's moved into a channel that contains the transcription factor, you can see that the force that's generated on the timescale of 80 to 90 seconds is approximately 0.5 piconewtons, which was consistent with our estimates from fluorescence. One thing that I thought would be interesting is that we could actually count the number of FOXA1 molecules within these condensates. And so the way that we did that was we actually looked at non-specifically bound FOXA1 molecules to the cover slip. And so essentially what happens is that some FOXA1 molecules bind individually, some bind as dimers non-specifically, some as trimers. And so we could actually bin a ton of these non-specifically bound molecules to the cover slip, and we could get an average intensity for a single molecule. And then we could simply count up the number, the fluorescence from within a protein DNA co-condensate, and then we could estimate the number of FOXA1 molecules from fluorescence. And what we found was that the average FOXA1 density within these protein DNA co-condensates was approximately 1,000 molecules per micron cubed. So if you think about the amount of condensation free energy being gained as being approximately 300 kBt per micron cubed, it's basically one FOXA1 molecule is contributing 0.3 kBt towards this condensation-based process. So we wanted to see how whether other transcription factors or other DNA binding proteins performed within this assay to see whether they would generate the same protein DNA co-condensation. So we purified Tata-box-binding protein, which is a general transcription factor. And what we observed was, in fact, that TBP formed small condensates along DNA, but it did not generate any protein DNA co-condensation, but rather performed more of a diffusive behavior. So this is the movie of TBP, of labeled TBP, and this is the chymograph along the DNA molecule. And here is the probability for TBP to condense DNA, and it's basically 0 for all end-to-end distances. However, when we looked at somatic linker histone, which is a structurally similar molecule to FOXA1 and has very known compacting activities, we found that somatic linker histone could condense DNA for all of the measured end-to-end distances. So the H1 condensates can very strongly condense DNA, which is again consistent with its known activity. So just to sum up, we found that these FOXA1 condensates can generate forces on the order of, so it's going back to these 3 questions that I posed at the beginning. FOXA1 can generate forces on the order of 0.5 piconewtons. We developed a theoretical framework that can quantitatively describe and estimate the coarse grain parameters associated with this process. And then from the protein biochemistry side, we found that the disordered C-terminus can largely drive this protein DNA co-condensation process, which is again consistent with other studies. And so, yeah, it's about 45 minutes. So I just like to acknowledge the Burgess Lab, where I did this work at both the MPI CPG and then the Max Planck Institute for the Physics of Complex Systems, the Grill and Hyman labs at the CPG, the protein facility, the LMF at the CPG, and also lots of interactions with nice people from the biological physics division at the BKS. Yeah, I'd be happy to take any questions. Thanks very much.
Thanks, Thomas. That was a very nice illustration of how to link theory and experiments to understand something more deeply. You can please ask questions in the chat or unmute and ask a question. I might start us off just as people warm up. You found when you looked at different transcription factors that, you know, sometimes you find things that are consistent with FOXA1, like the H1 histones, and other times not. And so, you know, I know FOXA1 and histones mostly in cancer ideas. And so what do you think that this means in terms of the role that these transcription factors are playing in perturbed DNA synthesis and transcription?
Yeah. So I think it's neat to think about like linker histone as an example. So linker histone is mostly associated with actually repressive contexts. So linker histone will bind to heterochromatin. It strongly binds to heterochromatin. It's supposed to turn off or be associated with things that turn off transcription. So it could be that these forces, so the collection of linker histone molecules that are generating this force, it could be that it has to be stronger in order to perform this inhibitory activity. Whereas FOXA1, for example, is generating, at least in this in vitro context, we still need to see what's really happening in vivo, but it's generating forces that are less compacting, but this still allows, so it's not gonna disallow other factors to actually engage with the condensate, but it's gonna bring DNA that's far apart together, but not so much that other factors can't actually bind and perform other activities.
Yeah, okay. I'll have to think about that a bit more, but yeah, thank you. Okay, so one of the first questions in the chat is, have you tested, well, thank you for the beautiful talk. Let me also read the compliment. Have you tested the effect of ionic strength and protein concentration on these parameters?
Yeah. So for sure, that's a great question. So the chemical interaction that's relevant for the data that I showed are strong electrostatic interactions. So you have plus charges on the transcription factor and very strong negative charge on the DNA polymer. And so those things are coming together, and that's the major interaction. So salt, of course, plays a big role. And so, but the main thing it does is it just changes the number of molecules that can bind to the DNA in vitro. And so essentially you need, and this is a cooperative effect. So you need a certain number of molecules to localize to the DNA before you can get this condensation pattern. So this cooperative effect is also associated with like first order phase transition. And so essentially what the salt does is it just tunes the number of molecules that can bind. And above a critical salt concentration, you won't get enough molecules binding to the DNA in order to get this protein DNA co-condensation. That's the main thing that it's doing. There was ionic strength and there was something else, protein concentration.
Protein concentration.
Yeah. So but it's the same thing. So protein, by changing the protein concentration that you're adding into the channel, that's simply changing the number of molecules that are going to bind to the DNA. And so if you go below a critical concentration, then there won't be enough molecules that'll bind, and then you won't get the condensation effect.
Okay. Second question says, beautiful work, thanks for the lecture. Are you looking into how these condensate dynamics change in the presence of histone PTMs, for example, oncohistone variants?
Yeah, so that's really cool. So we've started doing experiments. So this is all like completely in vitro. So looking at protein DNA interactions. And so a very natural extension to this work is to put reconstituted nucleosomal or reconstituted nucleosomes onto these in vitro DNA molecules. So we started doing that. And it's neat because when you can make these reconstituted nucleosomal arrays without PTMs, and the preliminary work that we have shows that if you have maybe 30 to 40 nucleosomes on this very long piece of DNA, you can also, at least with FOXA1, you can condense these nucleosomal arrays in the same tension-dependent manner. But the neat thing is, and I think that, for example, if you were to acetylate these reconstituted nucleosomes, then you would of course change the chemistry of these. So if you were to acetylate specific tails, which you can do, you would imagine that this would influence how FOXA1 or H1 was condensing or was doing specific things. And so that's something that we really want to do, and to basically look at, because what the PTMs are often doing is changing the charge. We know the charge is really important, so that's a very clear future direction. Yeah.
Great. Okay.
I imagine it would have quite an impact. I think it'll be a really nice perturbation, essentially.
Perfect. Well, looking forward to hearing more about that. Leon has a question for you. Let's get.
Thanks, Tom. I don't know a whole lot about transcription factors, but I think that they bind to specific sequences in the DNA. And you didn't say anything about those number of sequences in the DNA and whether different pieces, DNA from different sources, might have different properties with regard to this. So it's related to what Morgan was asking in terms of what's the connection between this also in the transcription of particular genes.
Yes, that's a really important question. So, so the first off, like defining the actual motive that a transcription factor binds to is often difficult because there's lots of them. If you look at the main motive that FOXA1 is purported to bind to, there are, I think, seven or eight of those motives along this lambda-phage DNA molecule. So they're there. But all of the data that I showed is non-specific binding. So we have a key experiment, which is that you can actually make a mutant protein that, so the DNA binding domain has a region that is responsible for sequence-specific binding. So that's to the actual motive, and non-sequence specific binding. And if you mutate the sequence-specific part of the DNA binding domain, you see the exact same thing. So for us, it was like very, I would say, clear that this data was all related to non-sequence specific binding of FOXA1 to the DNA. But the thing is, the sequence question is really important. And so how is there a way to increase the complexity of the assay in a way to observe sequence specific binding? And so I think one way of tackling that will be adding nucleosomal arrays. So the problem is you get such strong non-sequence specific binding because the negative charge in the DNA polymer is completely exposed. There's nothing else on the DNA molecule, and so FOXA1 can just bind everywhere. But in the nucleus, for example, you have DNA that's wrapped around histones. FOXA1 also has to compete with like 5,000 to 10,000 other proteins or different factors. I don't know the exact number of proteins, but a lot more proteins. And so these more subtle differences in, okay, FOXA1 wants to bind a little bit more to this region, become more pronounced when you have in the context of the actual nucleus. That would be the way that I understand it.
Okay, thanks.
Okay, thanks, Tom. And one last question. Mike Guevara didn't quite understand how droplets buffer the tension on the spider web.
Yeah, that's a cool question. So the way like from a biological perspective that I understand it is that you have it's slightly different, but the idea is that you have a liquid or a glue that binds to the spider cell. And then I think the way that people understand is that there's this Rayleigh plateau instability that happens along the spider silk. So this is what's giving rise to the number of like a bunch of small droplets along the silk. And essentially, when a piece of the spider silk actually breaks, for example, it'll be rapidly drawn into one or many of these small liquid droplets. And I think the way that the buffering means that even if you break a bunch of parts of the web, the tension, because you have the silk being drawn into these different droplets, will be the same. So you can think about, like, if you think about the extension of the total spider web as being the total amount of non-droplet based silk, the tension associated, so if I were to be a fly that were to run into the silk, I would experience the same tension whether I had an extension equal to 1 meter or 600 centimeters. Does that make sense?
Okay, well, I missed the last sentence that you said, but I think I'm just going to have a look. So, but is it just that the blob, I mean, because our tension is probably depends on the cross-sectional area of the fiber, of the strand.
Yes, that's a relevant, for sure. That's
so I was thinking it was just the fact that you had an increase in the cross-sectional area in a periodic way. Somehow that was good for the that would decrease the attention in a single strand. But let's not waste time on let's not waste too much time on that question. Okay, there are more important things that other people have to ask you.
Just one last thing. I think the key is to think about the silk as being either within a droplet or not, or as a substrate for droplets. That's the thing to think about.
Okay. It was good to see you again.
Yeah, you too, Michael. Thanks a lot.
Michael, all questions are important, so let's not make judgment about the importance of questions. I don't actually see any other questions in the chat, so I just want to say thank you again, Thomas, for such a nice talk and with great questions that came from it. Our next seminar will be the first Wednesday again in February. It'll be Simon Gravel, who works at McGill, so many of you may know him as well. And so I will send an announcement for that a couple of weeks in advance. So thanks very much, Thomas, and we hope to see you soon in Montreal.
Thank you. Yeah, it was really fun to give the talk. Thanks a lot.
Perfect. All right. Well, see everyone next time. Thanks.
Thanks, Morgan.
Thanks, Morgan.